Hi, now let's talk about stem cells and cell death. So the cells, from the beginning, the cells will differentiate into a terminal state, which is a specialized cells. And cell differentiation is a process of cellular specialization in function during an organ development or tissue repair and even normal cell turnover with the same genetic material. Especially quite recently, stem cells become of great interest to biomedical engineers. And stem cells are unspecialized precursor cells that can differentiate into a diverse specialized cell types, or it can self-renew. So for example, in developing embryo, stem cells can differentiate into 4 specialized cells. So that's great. And in adult organisms, like us, we are already adults. What are our stem cells? And stem cells acting as a repair system. So we know that we got some injuries, small cut, in a few days we will heal this, and over time it will even like disappear. So for our body and maintain our normal turnover of regenerative organs, such as blood, skin, or intestinal tissues. And stem cells are found in all multicellular organisms. So for us, like biomedical engineering, even out of curiosity, we are curious about how like our blood are keep going. And for example, red blood cell in our human body, they actually last for only 120 days, and then they will die. So we have to keep generating those new red blood cells inside our body. In fact, it's in the bone marrow. So this potential use of stem cell as a repopulating and tissue regeneration is of a great interest to us, especially when there's injury, there's an accident, a loss of tissue, or a problem in organ for replacement. So potential cell source as a stem cell is very important, and cell replacement therapy for diseased tissue, also for creating an engineered tissue, such as, let's say, skin substrate, skin replacement for burn patient. And as a seed cell, stem cell would be very interesting to us. So let's consider about the definition of stem cell. So last time we learned that in vivo fertilized egg and that zygote has the ability to become the whole cell types in a complete organism. So that we call as a totipotent stem cell. And once they divide into, let's say, for example, 8-cell embryo, and they will start to make a sac, which is called a blastula, an animal embryo at the early stage of development when it's hollow ball of cells. And that blastula now contains a small inner cell mass, and that is called the blastocyst. So it's a mammalian blastula in which some differentiation of cell has occurred. And that inner cell mass can become many types of the cells in our body. So we call it as a pluripotent stem cell that can be cultured and that can be differentiated into neural cells, that can become cardiac cells, that can become blood cells. Of course, there's a more differentiation so that particular levels of stem cell, which can only differentiate into one type of terminal cells, is called unipotent. So these term, toti, comes from Latin, the whole, and pluri, you remember that in English, a single versus a plural form. So that pluri means in Latin, very many. And this potency means having power to become any kind of cell types. So this potency definition of stem cells can be explained as a first, the stem cell which can construct a complete viable organism is called totipotent stem cell, or some people say this is omnipotent. So good example is a fertilized egg. And descendants of these totipotent stem cells, which can differentiate into almost any cells in our body, is called pluripotent stem cell. A good example is that embryonic stem cell. Embryonic stem cell, however, is coming from the placenta, so it cannot form placenta. Now, these pluripotent stem cells can and downwards, the stem cells can differentiate into a number of cells, but only those closely related family of cells we call as multipotent stem cell. A good example is hematopoietic stem cell, which is a blood cell making stem cell. And then the last part of the stem cell is the stem cell can differentiate only into one type, such as muscle cell or skin cell. So there exist these stem cells which can replenish our skin or a muscle. So that's called, because it can differentiate into only one type, it's called a unipotent stem cell. And now let's discuss what kind of stem cells are available. So first, isolated from an embryo, the most potent stem cell is called embryonic stem cell. But as you can imagine, embryonic stem cell is after this fertilized egg. So from the religious point of view, this can cause an ethical issue for stem cell research. So there's another breakthrough happens. We found an adult stem cell, which is from the tissue of an adult organism, such as you can take out a little bit of the skin of myself, and that if you can make a stem cell out of this, that can become called stem cell. Or for example, bone marrow or peripheral blood, and it contains stem cell, and that's called adult stem cell. Now, a revolution in the stem cell field happened not more than 10 years ago, a decade ago. It's called IPS, or induced pluripotent stem cell. By definition, induced and pluripotent is a very much capable, potent stem cell. And these are artificially derived from a non-pluripotent cell, typically adult, body cell. So it's an adult somatic cell. And that, that gave rise to a Nobel Prize in 2012. Do you know when the first paper on this came out? Majorly by Shinya Yamanaka from Japan. The paper in Cell was published in 2006. So in only about 6 years, he got Nobel Prize out of this discovery. So that means a lot, that is how important this field and how interesting this field is, because you can take out adult somatic cell and reprogram, means he has 4 factors of transcription factors into the cell and reprogram it to a pluripotent stem cell. So that resolves the ethical dilemma using embryonic stem cell. And so this is the wording in the Nobel Prize for the discovery that mature cells can be reprogrammed to become pluripotent. And you can ask questions like, okay, what are the stem cells? They are cells, so they may look similar. It's hard to find. So by definition, they exist in very small quantities. That's why it has been so long to discover the stem cell within tissues of all adult organs. So let's consider the characteristics of stem cell differentiation. So you look at this stem cell, which is quite interesting. Stem cells were divided into 2, and it can give rise to 2 stem cells, but it also possesses very interesting feature. So you see stem cells divide into the same one, so the self, and you look at this, it's just a cartoon, but it's a different one called committed progenitor cell. So it can divide into the same one, and that will give rise to more and more stem cell. That's called self-renewal. While this also gives rise to progenitor cell. What's progenitor cell? Means a cell which has a tendency to differentiate into a specific type of cell. And it's a push it to differentiate into that target cell. So you can look at these progenitor cells, then they will amplify. So it is called transit amplification stage, and then eventually they will become a terminally differentiated cell. So this very interesting property of the stem cell characteristic is divided into here. First, asymmetric cell division. And this part can self-renew for forever, in fact. So that's called limitless self-renewal, which is quite interesting. And also this multi-lineage of differentiation. And so all this property allows the stem cell to repopulate of tissues in the recipient or in our adult.